Stanford University.
Welcome to Stanford Medicine's Contemplation by Design Summit. I'm Dr. Tia Rich, the founder and director of Contemplation by Design. It's my great honor to introduce our speaker, Dr. Manus Donahue, who will be talking about meditation and the glymphatic system. Dr. Manus also has his MBA, and he is a professor of neurology within the Division of Cognitive and Behavioral Neurology at Vanderbilt Medical Center, and serves as Assistant Vice Chair for Research. Donahue trained at Duke University, earning his BS in physics and his BA in philosophy. He earned his PhD in biophysics at the Johns Hopkins School of Medicine, and then completed a postdoc in neurology at the University of Oxford. His work is focused on using new imaging and computational approaches to characterize tissue function in health and disease, and he has led NIH-funded trials whereby these approaches are applied to understand the influence of different behavioral states on brain function, and also applied in patients with atherosclerotic cerebrovascular disease, moyamoya disease and syndrome, sickle cell disease, and neurodegeneration to evaluate emerging therapies. Please join me in giving a warm welcome to Dr. Donahue.
Yes, thank you for that nice invitation. I appreciate it. And thank you to the organizers as well for inviting me to speak. And I'll go ahead and share my slides here. Right, so thank you again to the organizers. I hope everyone had an opportunity to go to the last talk as well. I enjoyed it. It was very nice. This one is going to be quite different, but hopefully you'll also find it useful. And it's going to be a little bit more focused on the neuroscience and the physiology of what goes on in some contemplative practices and with meditation. And hopefully, one of the things I like about these symposia is that you get different perspectives on similar problems from different people. And hopefully it provides kind of a more complete perspective on the overall field as well. So just again, I'm a professor of neurology at Vanderbilt Medical Center in Nashville, Tennessee. And what I'm going to be talking about today is meditation and the glymphatic system, which I'll talk a little bit about what that system is if you're unfamiliar with it. It's only been sort of identified for the last 10 or 12 years or so. Just to get started, these are my disclosures. We do a lot of clinical trials in our division, in our lab. None of these disclosures are relevant to the content of this work, with the possible exception that I'll say that the funding for this work was from the National Institutes of Health. And I wanted to start by just giving a short background on sort of where I came from and how I got to the point where I'm giving a talk on meditation in the glymphatic system late on a Wednesday evening. And my training started at Duke University, like it was mentioned before. And at that point, I was interested in physics and philosophy. And more specifically, on the philosophy side, I was really interested in ancient philosophy and metaphysics. And then on the physics side, quantum mechanics, things that are theoretical quantum mechanics, things that are sort of largely useless by a lot of current perspectives, I would say. But I was really interested in physics, among other things. And when I came to graduate, I thought, what am I going to do with a degree in physics that's going to be, worthwhile? And you can, there are different things you can do. You can go into business, or you can go into the military, you can make bombs and so forth. And what I thought, also based on my philosophy background, is that I thought going into medicine or biology would be a very noble application of physics. So, it would be a way to help people cure diseases, hopefully. But I knew nothing about biology or medicine when I graduated from university. So I went into it sort of purely on thinking that it was kind of a noble thing to do. And I went into a biophysics program at Johns Hopkins in Baltimore, Maryland. And at that point, I worked on developing new imaging methods, mostly with magnetic resonance imaging, which I'll talk a little bit about, to study how the brain works in cerebral physiology. But the work was really at this point still focused on developing new imaging methods. And what I always think, even to this day, is that there's a lot of problems in medicine that people have been working on for the last 50, 100 years or so, and we haven't solved those problems. And it's difficult to come up with new experiments or new ideas unless you have new methods. When you have new methods, you can measure something new, then it creates a whole new, world of opportunities for what you can do. So that was kind of how my training at the PhD level, anyways, progressed. But it was still a bit technical, developing methods. And then I went to Europe, which I really enjoyed. I've lived in Europe 3 times now. As a postdoctoral fellow, I was in a neurology department at Oxford, which is about an hour outside of London. And there I focused more on taking these new imaging methods that I've been working on developing with others and applying them to really start studying patients and looking at their applications in medicine. And the focus there was really in stroke. So those are sort of disruptions of the blood supply to the brain. And then also in neurodegeneration, so where the brain deteriorates. So things like Parkinson's disease, Alzheimer's disease, Huntington's disease. And I found that even more rewarding. And you can see that my trajectory is going from kind of being more technical to being a bit more applied by this point. And since about 2010, I've been a faculty member at Vanderbilt Medical Center, where I'm a professor of neurology and psychiatry. And we have a lab of about 20 people where we develop about 20% of the lab is focused on really developing new imaging methods and biomarkers that can be used to help understand brain function. And then now the majority of the work, I would say 80%, is really in taking those methods and applying them in clinical trials of different diseases or different treatments to understand how new, prospective treatments can affect brain health and improve, overall quality of life. And that's where we are currently. Just like I mentioned, we've got a lab. It's about 20 members. It's very multidisciplinary. So we have, very good clinical coordinators, very technical biomedical engineers and physicists. We also have neuroscientists that are more applied and medical professionals, especially throughout the fields of neurology, radiology, psychiatry, and so forth. And again, the goal here is really to use imaging, and specifically new imaging methods, to identify changes in tissue function that what I like to think precede irreversible tissue damage. And I'll talk about what that means a little bit. And if you can do that, if you can understand the brain health and specifically these changes in the brain that occur prior to the brain deteriorating to the point where there's no longer a point of intervention, then that presents a point where you can titrate therapies, whether those are pharmacological, surgical, or behavioral, which I'll talk a little bit about today, to patients with highest need. And you can do that at a stage before there's irreversible damage. So it has, potentially a large impact on patient health and whatnot. If you wait too long, then there's less you can do, obviously. And how do we do that? So there's different methods that we use and different approaches, but common to a lot of our studies is really magnetic resonance imaging. And as I mentioned before, I was trained a bit as an imaging scientist. For this talk, I will not go into the details of most of the imaging methods in too much detail. I will give you kind of a cursory view, but if people have questions about how they work, parameters and so forth, that's an area close to my heart. So feel free to ask those questions and I'll be happy to discuss them as well. What I do want to preface the work that I'm going to talk about with is it's just some general points about magnetic resonance imaging if you're unfamiliar. So first, it looks a bit similar to other scanners that you might see in the hospital, like a CT, a CAT scanner, or a PET scanner. But it's different in several key ways. And one is that it is considered relatively safe compared to a lot of those other modalities. And the reason for that is because of 2 points. One is that it's non-invasive. So we can get beautiful images of the body relatively quickly without any injections, which is different than a lot of other imaging modalities. Also, it doesn't require any ionizing radiation, things like X-rays and so forth. That makes it very good for things like surveillance, right? So if you want to scan somebody, you want to give a drug, have them meditate, look at how they sleep, and then scan them again, you can do that without worrying about dose restrictions or so forth. So it's good for, I always say, surveillance and looking at interventions. And then it's also, in addition, it's a very flexible and varied contrast. And what I mean by that is in a period of minutes, you can look at the blood vessels, You can look at the tissue itself, how much tissue is there. You can look at the fluid in the brain. You can look at iron. You get all of that different information without any injection. So what I always say is like it actually provides a large amount of information in minutes. And you might think, well, isn't it really expensive? It depends who you look or who you work with, but I would say that it is a good value relative to the information that you can get. In 30 minutes or so, you can get a wide amount of information, and you can really customize these approaches to look at very different components of physiology, which allows you to sort of test new hypotheses and so forth. And these are just some of the things that we look at. This is our protocol actually in stroke that we run now. And it takes about 30 minutes or so to get all of these images, but you can look at the blood vessels in the head. So this is the intracranial vasculature. You can also look at the blood vessels in the neck. So whether you have a stenosis in any of those vessels that might put you at risk for stroke. You can look at the tissue in the brain and specifically the white matter disease. So whether there are problems with scarring of the tissue or not enough blood getting to different regions. For this talk especially, I'm going to talk about the fluid. So you can also look at fluid in the brain that's bright on these images. You can look at the amount of tissue, such as the gray matter and the white matter. You can quantify that pretty accurately at sub-millimeter resolution now. You can look at things like diffusion. So this is how the water is moving in the brain. As it turns out, when you have a tumor or when you have a stroke especially, you'll get something called restricted diffusions where the cells will swell, and then you can identify that quickly in about 30 seconds on MRI and identify which parts of the brain are affected. You can measure the blood that's going to the brain and which vessels it's coming from, and you can do some fancier things with looking at how the vessels are able to respond to different stimuli and whatnot. And all of this information can be obtained in about 30 minutes or so. It's non-invasive, so you can look at it before and after an intervention or during an intervention. And again, I think it's a relatively good value for the information that you get. I will qualify that if you think that MRI is expensive, it's kind of an interesting side point here, is that if you were to run this protocol that I just listed here, which again takes about 30 minutes or so in a hospital, it would cost about $9,000. So I should say they would bill your insurance $9,000 for this protocol. But that's because there's a huge profit margin on these things. And the operating costs for a protocol like this are actually only a few $100s. So if you have your own scanner or if you have a research rate or something like that, you can get all of this information. Usually it costs $500, $600 or so. And it's not free, but it's in some ways a good value for the information that you get. So what do we do with that information? Well, what I've done here is I've taken a healthy person, and you're looking at their brain from the back. So this is a coronal representation. And on this side here, I've just shown a healthy person. And then on the other side, I've shown a patient with neurodegeneration. So this is a patient with Alzheimer's disease. And you can see that the two brains look very different, right? So you see a few things. One is that the sulci here, they become much deeper in the setting of neurodegeneration. And that's because you have tissue that is receding as a result of atrophy. And what the brain does is the size of the intracranial volume always remains the same. So what happens is when that tissue recedes away, that space will be filled in with fluid, which is dark on this image. So you can see that what's called the ventricles here in the middle of the brain, I'm going to talk a bit about those, that will expand in order to fill in the space that was receded by the tissue. We all probably have people that we know or have known that have had Alzheimer's disease. So it's a terrible condition. A lot of our work is focused on this, and we have some new drugs now that are have a high potential for improving cognitive impairment and reducing the burden a bit. But the problem with neurodegeneration, and as we age generally, is that it's very difficult, if not impossible, to reverse these changes. Once your brain starts to look like this, the tissue has died, we really don't have treatments to regenerate that tissue. So the work that we're really focused on is not necessarily identifying brains that look like this. We can do that without a fancy MRI scanner. But it's really identifying the changes that occur before your brain looks like this. When your brain still looks like this, but what are the subtle functional changes that are going on within your brain that are predisposing you to poorer outcomes later in life? And that can have a wide variety of possibilities. So you can have things like vasculopathy. So the blood vessels themselves become narrowed. You don't get enough blood, oxygen, glucose to the tissue. Also, we can look at things like chemistry. So you can look at Things like how much protein is in different parts of the brain, how much dopamine in this case is in different parts of the brain. You can look at the blood flow that's going to the tissue. You can look at the metabolism, how much glucose or oxygen is being consumed, how much iron is being retained in the brain, whether there are microbleeds, whether the vessels are rupturing and so forth in a small level. All of these changes that are occurring here are largely functional changes, and they can precede these sort of gross changes in tissue structure. So what we'd like to do is be able to identify, these functional changes that are occurring at a point where we can intervene. And what those interventions are can be different. And we have lots of those things that we study in our lab. But what I'm going to talk about today is meditation, actually, which is not something that I usually talk about. I should preface this as well by saying I've been in the meditation field for probably 3 or 4 years or so. So I know a lot more about cerebral physiology and brain health and imaging potentially than I do about meditation. So I am more of a novice in this space. I've learned a lot about this in the last several years and I've enjoyed it, but I would certainly be interested in hearing others' opinions on the studies that we're doing and the potential benefit as well. So from a neurology perspective, what are the treatments that we think about? And as people here know, probably the most common treatment that we have are pharmacological treatments, so drugs. So the neurologist that I work with, he always says, we got a drug for that, So this is kind of how I think in the clinic, most people probably think, or maybe also surgical techniques. So we work with different, especially supravascular techniques and seeing how they affect brain health. And these are probably the 2 most common treatments that are used, commonly in neurology. But we also have some other things like rehabilitation. So we're starting to do more things with like cognitive tasks, and maybe if you keep your mind sharp or so forth, you can prevent these sort of long-term changes, and other forms of physical rehabilitation and whatnot. But what is it that we actually don't use very much of in neurology from a clinical perspective anyways? And that are things like behavioral changes, meditation, right? And I'm talking to an audience today that I think probably has a lot of faith and interest in meditation and its impact on well-being. But on the clinical side, it is less utilized, certainly, and there's a wide variety of sort of perspectives on how useful it is. And those are some of the things that we were interested in investigating. And when people think about meditation on the neurology side, this is one of our participants here. They think about, things like this, and it's a little bit abstract and whatnot. And why is that, or what can we do to change those things? And the way that I view it, and there are different ways of viewing this, is that there's far more evidence for the impact of drugs and surgeries on objective health outcomes than there are things like behavioral changes like meditation. And that's because there are a lot of randomized controlled clinical trials that utilize established markers of efficacy. So that means you have a randomized trial, you bring in 100 patients, you give them a drug, 100 get placebo, and then you look at who lived longer. How do their cognitive functions change? What's the cerebral physiology? And there are a lot of, to be honest, very rigorous and good controlled randomized clinical trials looking at surgeries and pharmacological treatments. There are far fewer of those trials looking at the effect of meditation and whatnot. And not to say that there aren't any, there are certainly good trials on that. But I would say that there's a disproportionate amount of evidence that's more kind of anecdotal when we talk about the health benefits of meditation. And that's not meant to demean meditation in any way. It's just kind of the way that it's viewed on the clinical side. And what I think is that for meditation to be more accepted broadly among healthcare professionals, we should also have a focus on better understanding the mechanisms underlying these health improvements that people that meditate are known to have. And that's what we wanted to sort of start looking at with some of our imaging methods in the last several years or so. And we were contacted by the National Institutes of Health a few years ago. They were interested in using or having us apply for some funding to use our methods to look specifically at how fluid circulation in the brain changes during sleep and also during meditative states. And that's what I'm going to talk about today. And the background for this, I'll get into a little bit of the detail with this, But the high level background that I just want you to appreciate is that the brain partially mobilizes metabolites, proteins, waste products through the fluids in the brain, primarily the cerebral spinal fluid and the interstitial fluid. And I'll talk about what those are in the next few slides. There's a lot of evidence now coming out that sleep increases this mobilization of these waste products. So when you sleep, you upregulate these clearance pathways and you flush out all of the junk that's in your brain. And the saying that we have is that sleep is the price you pay for being awake, right? And that's when your brain kind of cleanses itself for the most part. But we actually don't know a lot about sleep as well, I would say. If you think about it, from an evolutionary perspective, the radiologist that we work with always says this. He goes, a long time ago, sleep, puts you at great risk, right? Like if you sleep, you could be eaten by a lion, you're not observant. So the fact that this has been preserved, over the period of evolution and so forth, means that it must have, really very relevant physiological implications, which we know. And the hypothesis that we had here is that focused awareness meditation, mindfulness meditation, and I'll get into what we did exactly, mimics this mild sleep state. And what it may do, among other things, is mobilize the clearance of these cerebral peptides and waste products. And if it does that, it could have similar restorative effects as sleep. So that's the overarching hypothesis that we're looking to test. And we're going to do that using non-invasive MRI methods that are sensitized to fluid motion and to brain activity during different behavioral states. Okay. So a little bit of a background on the fluid that's in the brain. And I'm showing a video kind of scrolling through the brain here. And these are the bright regions on here are all the fluid in the brain. And I'm going to talk a little bit about where this fluid comes from and where it goes. The fluid that I'm mostly going to be talking about is cerebral spinal fluid, or I'll abbreviate that CSF. That's all of the white fluid that's here. And it has lots of roles. So initially, we always thought it was important for protection, so it kind of prevents some buoyancy and whatnot. But we also know that there are certain nutrients and waste products that circulate through this fluid. It's produced within the brain, and then it can flow out of the brain into the blood. The brain generates approximately 500 milliliters of this cerebral spinal fluid, or CSF, every day. But within the brain, there's only about 125 milliliters of that fluid. So what that means is that this fluid is constantly circulating. And the circulation of this fluid is really fundamental to homeostasis. When the fluid doesn't circulate correctly, gets stuck within the brain, you can have conditions called like hydrocephalus and so forth. And that can put pressure on the brain. You can die from that. You get symptoms, puts pressure on the tissue. You don't clear your waste products effectively. And those are very serious conditions where you have to shunt the brain to pull out that fluid or so forth. But we now know that this fluid circulates during sleep, or it increases its circulation during sleep. It also circulates during the day. So what we're going to look at here is whether we can modulate this fluid, not just with sleep, but also with focused attention meditation. Okay, so a little bit more detail on how this fluid moves throughout the brain. And what I'm showing here is a single person, and you're looking at their brain from the sides. That's the sagittal image, cross-sectionally, so that's the axial image in the middle, and then coronally, looking at it from the back. And everything that's bright on here is this cerebral spinal fluid. And a couple regions that I'll just draw your attention to is that you've got 4 ventricles in your brain. That's where most of the fluid is produced. And you're looking at the lateral ventricles here in blue. Those are kind of up midbrain here, or in the middle of the brain, I should say. And within all 4 ventricles of the brain, you have these cells, this cluster of cells that are called the choroid plexus. And that's what actually produces the CSF. We kind of think about it, some people think about it anyways, as kind of the most proximal component of the CSF circulation. It's the beginning of it. It's where the blood plasma comes up, it's filtered through those cells, and it creates this fluid. After that fluid is created, kind of most of it is created in the lateral ventricles in the middle of the brain. After it's created, it's going to flow down through this little pipe, which is called the cerebral aqueduct. So it's kind of easier to see this on the coronal view on the right. So the fluid's produced in the ventricles here. After it's produced, it flows down through this pipe called the cerebral aqueduct. Once it does that, it's going to circulate around the brain, and it's going to go up to the top of the brain, and it's going to move into the veins, into the blood circulation through something called arachnoid granulations. So the fluid is produced in the choroid plexus complexes, flows down through the cerebral aqueduct, circulates around the brain into these arachnoid granulations, and is cleared out through the blood. That's called the sort of bulk CSF flow pathway, which we've always known about. And I'm going to talk a lot today about flow of this CSF through this cerebral aqueduct, this little pipe. We've known about that pathway for quite a long time. So this is not controversial. And this is always how we've thought that fluid circulates around the brain. And just as a reminder, produced in the choroid plexus of the lateral ventricles, flows down through the cerebral aqueduct, circulates around the brain, and is taken up through these arachnoid granulations. And that all happens. It's happening in all of us right now. An extension on this model of how the fluid circulates within the brain happened about 12 years ago or so now, where this glial lymphatic or glymphatic system was proposed. And what this is if you zoom, in addition to everything that I just said, which all happens, if you zoom in to the brain tissue or the brain parenchyma, and you look at what it looks like, this is actually from a chinchilla, so mammalian scan and electron microscopy, but our brain looks similar. You have blood vessels that are in the arterioles. They have this smooth muscle around them that allows them to expand and contract. And then you also have the veins, which kind of where the blood flows out of. And what was identified is that if you look around these blood vessels, there's also fluid that moves around those blood vessels. And the motion of that fluid is very sensitive to how those vessels are moving. So what the pulsatility is. Are they moving quickly or slowly? They're not compliant. That motion won't go quite as well. And I've just drawn a cartoon of what this looks like. And so if you look at the artery, which I've kind of shown here in the red, right around that artery, there's this what's called perivascular space, which we now know. And this is fluid that's moving right around that vessel. And it's stuck in that space because there are these astrocytic end feet, so brain cells that border that space on the other side. And that fluid and all the waste products that are in it will kind of move along those vessels. And then ultimately it can move into this interstitial space. So it can get through these astrocytic end feet, usually through aquaporin 4 channels, so water channels. And then it moves interstitially. So no longer in the perivascular space, but kind of moving around the astrocytes and so forth. And it'll find its way to a vein, and it'll move into the perivascular space in the vein. And then it will flow down. And we don't know exactly what happens to it yet when it flows down, but it probably ends up in the neck and some of the lymph nodes that process waste products in the neck. That's still not well known. But what I want everyone to take away from this slide is that there's a new clearance pathway within the brain that's recently been developed in the last decade or so, or identified, I should say. And fundamental to this clearance pathway is the sort of motion of fluid around the blood vessels and through this interstitial space. And if you think about a lot of the problems in the brain that we have, they're what's called proteinopathies. And what that means is that there's too much protein, waste products that are accumulated in the brain. Alzheimer's disease, it's something called beta amyloid plaques. Parkinson's disease, you have alpha-synuclein. Huntington's disease, you have Huntington protein. Multiple sclerosis, you can have things accumulated in the interstitial space. All of these conditions that we really don't know well what causes them or how to cure those conditions. There's now a lot of focus that this glymphatic system could be a major player in what's going on in these neurodegeneration conditions. And basically what's happening is that you're getting these waste products and solutes stuck either along these perivascular spaces around the arteries or in these interstitial spaces because something is going wrong with how this pathway is being regulated. And I've just shown a cartoon here and kind of how we think about this. So you can kind of have a healthy person looking cross-sectionally through the artery here and the veins. This is kind of a 3D version of what I just showed on the last slide. Everything kind of comes through nicely, goes into the interstitial space, into the venous perivascular space, flows out. But it's possible that this goes wrong where, maybe the arteries aren't pumping as well. So the arterial blood volume is now smaller. So that means there's a lot of this junk, these solutes that are getting stuck in this perivascular space. Or maybe there's blood products that are in there and so forth. And then you can't get things flowing along there. Maybe there's some problem with what's going on in the interstitial space. how the brain activity is actually regulating this movement interstitially. And that's done with, with neuronal and astrocytic activity. Maybe that's causing things to stagnate. And all of these problems, you could get retention of these waste products within the brain, and it could lead to some of these neurodegenerative conditions. So what you can see with this is that this is fundamentally dependent on the vascular tone, how compliant these vessels are, because all of this is kind of moving along with the vessels, and also the brain activity itself, how these cells are working, because the idea is that a lot of this stuff flows interstitially as a result of convective currents from astrocytes, neurons, and so forth. So if you can modulate the vascular tone, or you can modulate the neuronal or the astrocytic activity, then you could potentially help facilitate improved clearance of these waste products. And that's a lot of what we're focusing on doing right now. And it's difficult to do that. It's difficult to modulate these things with drugs and whatnot. So can you modulate it with behavioral state? And this is what was initially proposed by the person that identified this glymphatic system, Megan Niedergaard. This is a more recent paper, actually, but what she's been doing is really looking at how lots of things, but one of the things is really looking at how this system is modulated by behavioral state, especially in animals. So what you can do in animals is you can take a mouse and you can just take some dye or some fluorescent tracer or something that you can track with imaging, and you can inject it straight into these CSF spaces, straight into where the cerebrospinal fluid is. And what she did here, she did this near the bottom of the brain in a fluid-filled sac. And she did that in awake mice. And then what she did in the bottom here, she looked at what happened to that tracer within the brain. And it kind of is retained more or less where they inject it. You don't see it going too many other places. When the mouse sleeps, you can look up this paper. They have nice videos of mice sleeping while they're catheterized with this stuff. It's kind of interesting. You can inject this stuff into the cisterna magna here. And when they're sleeping, you get a very different response. So no longer does the tracer just stay where it's injected, but now it moves throughout the brain. And specifically, if you look at the other images in the paper, along the blood vessels, the middle cerebral arteries, some of the big blood vessels that perfuse the front half of the brain, front 2/3 of the brain. And interestingly, if you anesthetize the mice, you see something similar as well. That also seems to mobilize some of this. So it's work that's showing that the circulation along these perivascular spaces, potentially these glymphatic channels, increases when you sleep. And the thought here is that you're suppressing the neuronal activity a little bit, you're changing the vascular tone a little bit, and it provides just the right combination of effects to move this fluid through more coherently. Now, the problem with this, as you can imagine, is it's difficult to inject tracers straight into the brain of people. So what we've been doing and others is working on developing methods where we can make similar measurements without having to do any injections. And magnetic resonance imaging is really where a lot of this field is going. This is work that I think is quite seminal. It's not from us, so this is from a group in Boston, and I put the citation here. And what they looked at is specifically how fluid is flowing into the fourth ventricle down here at the bottom of the brain, at the same time as how the blood flow and the blood oxygenation in the brain are being modulated. And they did this with a technique called the Bold effect, which is the blood oxygenation level dependent effect. And if people have questions about that, we can talk about it later. But what I'm going to focus on just for this sort of background is that they looked at how the signal, again, in the fluid is relating to the signal in the brain and around the blood vessels and so forth. And if you look at someone while they're awake, and you look at the signal in the ventricles in the fluid, it looks something like this here in purple on the bottom. And then if you look at that signal in the tissue, which is really the blood flow, the blood oxygenation modulations that are going on, it looks something like this. And this is when the person's awake. When they sleep, and they confirm sleep using an EEG compatible cap where they can do EEG measurements, what they saw when they sleep is something very different. And they did some filtering here, which makes it also look different. But what they see is that in the fluid, about every 20 seconds, there's this huge peak, and then it goes back down. And it's very different than what you see when you're awake. And when that fluid signal peaks, when you get a big burst of fluid changes about every 20 seconds, the activity within the brain, the hemodynamic activity, drops. And you can see that that's very tightly anti-correlated, right? So what they're showing here is that when you fall into a deep sleep, this is a stage 2 non-REM sleep, about every 20 seconds, your brain has this pulsation to flush out the fluid. And when that happens, the activity within the brain suppresses, okay? Right, so our hypothesis is, can we measure something similar to that in mindfulness meditation? And we have a few methods. We're going to use that same method, but we have a few other methods that are a little bit more quantitative as well that we look at. One is that we can measure the velocity and the motion of this fluid going through the cerebral aqueduct as it flows from the ventricles down to the bottom of the brain. This is with a method called phase contrast angiography. It takes about 4 to 5 minutes. And I just want to show you what it looks like in 2 people here. And what you're looking at it here on the vertical axis is the flow of that fluid versus time, and it's very tightly correlated with the cardiac cycle. So we measure the cardiac cycle while they're in the scanner with ECG electrodes. And when the heart's not pumping, okay, so when the heart's not pumping during diastole, that fluid actually goes in a retrograde motion. It goes up towards the brain. And then when the heart is pumping during systole, it goes down. And overall, there's a net downward effect. But what you see if you look at a young adult who's like 22 years old, that's the blue curve here. The retrograde flow at the bottom, at the top, it's kind of small, and then also the flow down is also kind of small, and it's kind of tight. There's not a lot of kind of turbulence that's going on. And as you get older, what happens is it becomes more hyperdynamic. So when the heart's not pumping, the fluid's going up in the brain more. When the heart is pumping, it's going down more. And if you look at the difference between those two, the actual net flow drops a little bit. You got a little bit less fluid that's going out of the brain. And we've measured this now in more than 100 people, actually. But what you see is just kind of like what I said. The net flow of the fluid out of the brain, it's highest when you're younger, and then it starts to drop as you get older. So that's one thing that could potentially contribute with some of the problems that happen when you get older. And you can also make movies of this. So I just kind of showed what this looks like. Hopefully these come across okay over the internet. But this is a healthy person here on the left. And I've sped this up about tenfold. We get an image every 20 milliseconds or so. And you can kind of see how the brain's moving when you're awake. And you can kind of see it moves a bit with the respiratory rate and whatnot. And we're kind of really focusing on the cerebral aqueduct here in the middle. Suppose you look at someone that has Alzheimer's disease or they have Parkinson's disease. So first thing that you'll notice is there's a lot more fluid in the brain, right? So as the tissue recedes, that space is filled up by the fluid. And if you look at how the brain struggles over the respiration cycle and whatnot, there's a lot more struggle that goes on in the setting of neurodegeneration than there is during health. So there's a lot more hyperdynamic activity that's occurring when you get older and when you get neurodegeneration. So the question is, can we suppress that in some way? Can we control it? Can we improve the efficiency of the fluid motion? Because if we can, then you might be able to help generate these restorative effects of sleep and clearance just by behavioral changes. Okay, so that's looking at fluid through the aqueduct. And the last method that I'll just mention that we use is a way to look at perfusion within the brain, and that's the blood flow that's going to the tissue. And the brain is very clever about making sure that it gets just the right amount of blood to the tissue. And if that drops a little bit, it will dilate the vessels to get more, and it's very tightly controlled. And it also provides a surrogate of the activity of the neuronal activity. So the neurons are working harder, they need more blood, and you'll see increases in the blood flow. So we have a method of doing this called arterial spin labeling. You can do it all non-invasively. For the imaging people that are out there, what you do is you can magnetically label the blood water that's in the cervical vessels using a radio frequency pulse. You then wait some time for that blood to flow into the tissue, and it'll lower the tissue signal just a little bit. And then if you get another image where you don't label the blood and you do the subtraction, then the only thing that's left is the perfusion, where the blood is gone in the tissue. And it looks something like this. So you get this nice kind of blurry perfusion map in healthy people, which means the blood is kind of going everywhere in the right amount. The gray matter has a blood flow that's about 2.5 times higher than the white matter. So that's why it looks darker there. What you also might see, if you remember from what I said earlier, is in the back of the lateral ventricles here, you see this hyperintense signal, and that's where the choroid plexus is. So those are the cells that are producing this fluid. So what we've kind of thought for a while is that, if you can measure the perfusion, the blood that's going to those choroid plexus cells, then it kind of provides a surrogate of how hard those cells are working and how much fluid is being produced. That ends up not being entirely true, but it's roughly true. So if you look at the net CSF flow down through the aqueduct, how much fluid is kind of being produced and flowing out, that's roughly correlated with the choroid plexus perfusion, how much blood is going to the choroid plexus. But it's not great, right? Like there's quite a lot of variation in that. And what that probably means is that the choroid plexus is doing something more complicated than just producing CSF, which we now know. Also, if you look at how this perfusion changes with age, it drops with age. So as you get older, the perfusion to your choroid plexus in your brain generally, unfortunately, drops. Okay, so we have these methods for measuring the hemodynamic activity, the blood flow within the brain. We can also measure the CSF flow through the aqueduct. So what we want to do is see how these things change during sleep and meditation and so forth. And here's what we did. And this is a bit of an ongoing study as well, I should say. So we enrolled healthy adult participants. The age range right now is 18 to 55 years. And what we wanted people to meditate in the scanner. So we tried to recruit very adept meditators, so people that have lots of experience. And we had some criteria for doing this. So the estimated lifetime formal meditation experience had to be greater than 111 hours. It's close to about 1,000 hours in most of the people. They had these self-reported proficiency scores that we required. There was minimum expertise included, consistent with daily practice of 4 times per week, 20 minutes per day for at least 1 year and 1 retreat. Some other criteria that we imposed. Really, the goal of doing this is to get people that were really adept meditators. They do it every day with the idea that when they go in the noisy scanner and they're hooked up to a bunch of stuff, they can still do it. And that's what we aimed for. What we did here was a focused attention experiment, and I'll talk about that in a second. So we recruited from several different traditions and whatnot. People always ask, did we look at different traditions? Have we recruited other people? The answer is not yet. That would be interesting to do. I'd be interested in hearing feedback on that. But so far, we did a very basic focused awareness, mindfulness meditation study. During the study, we made some basic measurements. So we looked at like the tissue volume and so forth with MRI. We looked at the CSF flow down through the aqueduct. We looked at the activity of the choroid plexus with this perfusion measurement that I talked about, and also the perfusion or the blood flow into the tissue. We made the same measurements that the Boston group did during sleep, where we looked at the activity in the brain parenchyma, as well as the inflow of the fluid in the ventricles. And then we also did a whole bunch of clinical scans as well, just to make sure that nobody had any strokes or tumors or so forth that could also bias the results. And the task that we gave them, this is exactly what we read to them. So I'm just going to read it here as well. I feel like this is the slide that people may understand most and more than me. So I want to go through it so that everyone can appreciate it and provide feedback as is appropriate. We tell them, we're going to start the meditation scan, and please try to remain still and stay on task. Keep your eyes closed and try not to fall asleep. There will be approximately 20 minutes duration for you to enter and maintain a focused attention meditative state. We ask that you concentrate on the breath in the traditional Anapanasati style of breath meditation. Gently rest your awareness around the nostrils of the nose and upper lip as your focus of attention. Simply notice the sensation of the breath as it moves in and out of your body automatically and effortlessly. Don't try to manipulate your breath in any way, and let the breathing go on comfortably and stay with the sensation. The mind will naturally wander away from the sensation of the breath. Simply recognize this tendency, and with self-kindness and ease, bring your awareness back to the breath and maintain alert and focused concentration. We have them practice this outside the scanner. We also have audio recordings of what the scanner sounds like, so they can take these home and listen to them and potentially get used to it. And this is sort of the instructions that we give them for the scanner. Okay, and then the protocol itself looks a bit like this. So we bring people in the first day, and we make reproducibility measurements. And the reason I did that is because I thought the changes that we see with meditation are probably going to be smaller than what we see with sleep. And I don't know whether they're going to be beyond sort of a normal noise measurement for these measurements. So we bring people in, don't have them meditate, make all the measurements, and then make them all again in the same behavioral state. No sleep, no meditation, just seeing what the normal experimental error is in that measurement. We then bring them in and we do before and during this focused attention. So we do 25 minutes, similar to this, mind wandering, awake, no meditation, no focused awareness. And then right after that, we read them the instructions. We repeat all the measurements over the next 25 minutes. In addition, we have people that come in and we look at sleep. So they come in and one day we do 25 minutes where we make all these measurements. They're then sleep deprived, so they have to stay up all night. And they come in the next day and we make all the measurements in the sleep deprived state. That's kind of interesting. And then they're allowed to fall asleep and we make measurements for about the next 2 to 3 hours, or as long as we can keep them in there. With this, we use EEG caps to make sure to monitor the electrical activity and to make sure that we can record what sleep state they're in. We have a sleep neurologist that grades those to grade the sleep stages. Okay, this is what it looks like. So I always find this interesting. So this is when people come in, they're well rested, they get the stuff put on, they're kind of excited because there's lots of different things they get hooked up to. You then keep them up for a day and they look like this, right? And the question is always, what goes on in the brain that makes you go from this to this with sleep deprivation? That's what sleep does, right? And we always, we always say that sleep is the price you pay for being awake. Okay. Small aside, you might be thinking, if you've done EEG measurements in the MRI scanner, is it safe to do that? And we actually don't know. So a lot of the commercially available EEG equipment has been tested for very basic MRI scans, but not for a lot of the fancier things that we were doing here. So before we did any of this, we made some measurements where you can, it turns out that a ripe watermelon, interestingly, has about the same mass as a head and density as a head as well. And it provides a good sort of phantom to check the temperature and whatnot. So we would buy these watermelons, put the cap on it, and then we had this optical temperature probe where we can make temperature measurements in the scanner while we did all this. And what you see, this plot on the right here is the temperature that we measure versus time for lots of different sequences that we run and different electrodes that we've tested this for. And what you see is that the temperature doesn't change too much over the period of about an hour. It might go up by about 1 degree Celsius or so. So it's largely safe to run these sequences. We haven't had any adverse events or serious adverse events anyways with this stuff. So it kind of provides a little background that if other people want to do similar things, it's probably safe to do it as well. Took us a long time to get all this going, I think, so we always took pictures of this. So this is kind of day 0. This is one of our good graduate students in the lab unpacking all the equipment and figuring out how to use it. It took us a while to do that. We then get better and we're making the measurements, positioning things correctly, confirming the temperature, and then we do the first participant scans and so forth, testing in the scanner, fooling around and so forth. But over the period of a year or so, this probably took, we kind of got the protocol developed and all of the safety things done, and that's all done now and published. What these look like, if you look at the EEGs outside the scanner during a wakefulness, it might look like something like this. These are traces from different electrodes. When you sleep, outside the scanner, it looks pretty nice. You can get these nice, what's called K complexes that indicates sort of a stage 2 non-REM sleep or a deeper sleep. And then when you put the person in the scanner, it doesn't look quite as good, but you can still identify these K complexes. And again, we have a sleep neurologist who's used to looking at these things that grades all of the sleep. Okay, so what does it look like? So the first thing that I'm going to show you here is if you look at that absolute CSF flow, so that's how much motion of the fluid is going on through this cerebral aqueduct over the cardiac cycle. So it's how much is going down plus how much is going back, how dynamic it is. If you look at the CSF movement for repeated measures in the same state, so when we didn't have people do anything, you get pretty much the same number, right? That's what we wanted to see. If you then have them sleep, so you measure wakefulness in blue versus sleep, it looks like this. It's now the error bars are larger, and it's, as it turns out, it's difficult to sleep in the scanner. People do a different, some people are good, some are bad. But you see that effect that we kind of thought, right? Like as you sleep, the you don't see as much of this hyperdynamic CSF flow that you see in old age and neurodegeneration. It kind of has this nice restorative effect potentially. And then what's new, this is kind of known, and then what's new is what if you do the mind wandering that first 25 minutes, and then you do the meditation. And when you do that, you also see a significant reduction in this absolute CSF motion. So this provides some evidence that when you do this focused awareness meditation, the absolute CSF flow becomes less hyperdynamic, less coherent, similar as what happens during sleep. Right, and so that's what I said. So one of the things I started noticing in doing this, and people here are probably well aware of this, but when you meditate, lots of things happen. And one of the things that happens is your respiration changes, right? So you drop your respiration a little bit. And we measured that as well. So we hooked these people up to respiratory bellows. You can measure the respiration rate. And what we found is that your respiration rate actually drops from about 13 breaths per minute to about 10.5 breaths per minute in the people that we scanned anyways. And it's actually well known that fluid flow within your brain is very closely related to the respiration cycle. So I was a little bit concerned about that because I thought, well, maybe all that we're seeing is a respiration effect, which still could be interesting because that could be a point of intervention. So we enrolled another group of people, 13 of them, where we brought them in. There were people like me that were not meditation experts, but were really good at getting on the scanner and could control our breaths pretty well. And what we did there is we had people try to drop their respiration rate. They practice outside the scanner, but don't meditate or anything. So they dropped it from about 14.5 to 9.8. So it's actually a larger drop that we had them do here, not deliberately, than what we saw in the meditation. And when you do that, you look at this absolute CSF motion, you actually don't see the difference, right? It's kind of interesting. So I thought we would actually see the same effect. But when you do these respiration controls, where you're just dropping the respiration, but you're not doing any meditation, you actually see, you don't see that same effect. So it might mean that, could mean different things. But the interesting thing is it could mean that the meditation and the focused awareness is doing something more to the fluid motion within the brain than just a respiratory effect. Okay, so you look at more sort of detailed things that might be underlying this, and we can look at the velocity of that fluid that's going down when the heart's pumping, and then also the velocity when it's going back up when during diastole, when the heart's not pumping. And it's interesting with this, if you look at that maximum CSF velocity, first in these repeated same measurements, then in awake versus sleep, then in mind wandering versus meditation, none of these differences are actually significant. So they're all kind of within error, I would say. If you look at what happens during diastole, so when the heart's not pumping, and you get this retrograde flow going back up to the top of the head, if you look at the repeated measurements, get the same thing, that's good. Awake versus sleep, we also interestingly get the same thing, at least in our participants. And if you look at the meditators, you see that retrograde flow actually seems to drop just a little bit. So if you believe it all, what it provides some evidence for is that this hyperdynamic CSF flow reduces when you meditate, when you do this focused awareness meditation. And that's largely driven not by how much fluid is going down the aqueduct, but by how much retrograde flow is going back up. And there's less of that retrograde flow that's going back up. So that makes it kind of a more efficient process to clear the fluid potentially. Okay. Right. And then, we've, in addition to these studies, just to kind of corroborate these findings and think about it again, remember that the CSF flow, it increases with age, this absolute turbulence or hyperdynamic activity. So we know that's probably a bad thing. As you get older, you become more hyperdynamic in your flow. And what we see with the meditation is you go, you leftward shift on this graph. You go, you drop your absolute flow a little bit more like a young, healthy person. Right. And then, another thing to corroborate it, I always think we do lots of studies, and if it all kind of makes sense with different studies, then it's kind of nice. So Hett, who's a soon-to-be faculty member and senior staff member in the lab, he's been looking at patients with Huntington's disease for a long time. And these are patients that have neurodegeneration. It's also a genetically well-characterized, terrible condition, but it affects younger people generally. And what he found when you look at this fluid motion is that in healthy controls, the peak velocity of the fluid down the aqueduct is around here. And then if you look at people that are at risk for getting Huntington's disease, they will get it, but they're not symptomatic yet. That fluid has already increased. And then those people that are manifest, so they're in later stages, it's also increased. Again, a condition where neurodegeneration, the brain is deteriorating, and this fluid becomes more hyperdynamic. And again, when you meditate, if the results are correct, you shift sort of leftward on these curves. This is just another way of looking at that. If you look at this kind of flow across the cardiac cycle, you look at this maximal caudal flow, so that's what's going down the aqueduct. As the disease increases, that goes up. And then if you look at the cranial flow going down, as the disease increases, that goes down. So again, evidence that the flow becomes more hyperdynamic as the disease becomes worse. And again, when we, for the meditation and the sleep data, it seems that as you go into those behavioral states, you would shift a little bit left on these curves. Okay, so what about the other measurements? Well, you can look at that choroid plexus activity as well with the perfusions, or how hard is the choroid plexus working? Is that changing? You might think, well, that's a pretty small structure. How do you segment that, or how do you identify it? And for the last several years, we've also been working on developing deep learning algorithms where you can take the anatomical images, automatically pull out the choroid plexus. We validated that against gold standard tracings from a radiologist. This is now a tool that you can just feed in an image to, and it'll automatically pull out that information. So it's kind of nice if you want to take something like that and apply it to, like, a whole bunch of data sets from meditators or from Alzheimer's patients or so forth. And again, I showed this plot earlier, but what you see when you do something like this is that the net CSF flow, it increases generally with the choroid plexus perfusion, although it's a rough correlation. Okay, so what happens when you sleep and you meditate? Well, it's kind of interesting. If you look at the gray matter perfusion, repeated measurements, awake versus sleep, mind wandering versus meditation, we actually didn't see across the whole brain anyways, any change in the gray matter perfusion between these different states. If you look in different regions of the brain, that can change a little bit, but across the whole brain, we didn't see any change that was significant anyways. And if you look in the choroid plexus specifically, so my thought was that this perfusion would increase during meditation. You don't really see that. Maybe you can convince yourself it goes up a little bit, but so far it's not significant. So that effect is a little bit small if it's there at all. So that was something that wasn't confirmed, I would say. And then the last thing is, suppose you repeat this study that was done by the Boston group during sleep. And remember what they did is they looked at the fluid that's moving up into the fourth ventricle here at the same time as they looked at the hemodynamic activity in the brain. And so now we're going to do that same thing, but we're going to do mind wandering on the left, and then we're going to do the focus attention meditation. This was done by a talented undergraduate in the lab, actually, Bryce Keating. And you can see, you don't need any fancy analysis, that when you're mind wandering for the first 5 minutes, the signal looks something like this. And as soon as you meditate, you go into the focused awareness, it looks very different. Okay? And you can see that the amplitude changes a little bit, and also the frequencies change a little bit. So you can do something called a Fourier transform, which is going to take that information and it will figure out which frequencies are actually different. And if you look in the fourth ventricle, we also looked at the fluid around the spinal column, I should say. And when you do this and you look at all the different frequencies in that time course, are any of them different between meditation and mind wandering? So the meditation is in blue. And what you see is that around 0.1 hertz or so, and you see that in about the same region, both in the fourth ventricle and spinal column, there is a change in the frequency of the inflow into the fourth ventricle in meditation versus mind wandering. That frequency is a little bit different than what they found during sleep. So they found a frequency of about 0.05 to 0.08 hertz in sleep. We're finding something around 0.1 hertz in meditation. But if you think of, if you do the math in your head, that's still like 9 to 10 seconds per cycle. And that's well beyond like what a cardiac fluctuation would be. And it's probably, beyond what a respiratory frequency would be as well. So, the idea is that, maybe once every 10 seconds or so, you're getting, this pulsation of the CSF in the fourth ventricle that occurs during meditation, but doesn't occur during just normal mind wandering. And then what they also did is they looked at like how those pulsations are related to the hemodynamic activity in the brain. And remember, they found that to be anti-correlated. So we do that as well for the mind wandering and the meditation state. And what I'm showing here, if you look at the meditation, is in the spinal column. That's the fluid that's moving up and down. That's in blue, and it pulses by a lot relative to the, we looked here in the salience network, kind of anterior insula and so forth, which is relevant to meditation among other reasons. And we looked at the hemodynamic activity in that region. And if you zoom in a bit, on some people, but not all, you can stare at this for a while and you can convince yourself that there's an anti-correlation here. So if you look in the spinal column, the blue goes down here, and then the salience network goes up. Here, fluid's going up, the network's going down, up, down, down, up. So you can see as you go along here that in this person, it does look anti-correlated, and we see that kind of evidence of suppression of hemodynamic activity, which also correlates with an increase in the fluid motion during meditation. But I would say that this does not occur in everybody. It occurs in about 25% of all the people that we scanned, and then the others are a little bit less clear, and some of them are, the effect is not there at all. So it's something that on average, we found not to be significant, but we did find people that have effects like this where it seems very real. Yeah, so I just wanted to stop after a couple discussion slides here, and I'll kind of bring everyone back to kind of what the main findings are that we found so far. And just as a reminder, the goal here was to understand how neurofluid movement changes with aging and neurodegeneration. And the immediate hypothesis is that neurofluid movement and associated cerebral waste clearance can be modulated by sleep and focused attention meditation. The directionality there is similar. So prior work from us and others has shown that with increasing age, the cerebrospinal fluid, the fluid that's in your brain, flows through the aqueduct, and that becomes hyperdynamic with age. As you get older, there's more turbulent motion and so forth. This effect is also enhanced in the setting of neurodegeneration, Alzheimer's disease, Parkinson's disease, Huntington's disease. In the setting of mindfulness meditation and sleep, what we found some evidence for is that the CSF flow through the aqueduct, it becomes less dynamic during sleep, and then it also becomes less dynamic during meditation. And again, this is just a focused awareness meditation. This may be explained by similar systolic flow, so similar flow down the aqueduct when the heart's beating, but lower retrograde flow during diastole. So overall, it creates a more coherent pattern. This effect was not observed when we just did that respiratory control, where we brought in people that didn't do the focused awareness, but did modulate their respiration. And what I also want to say about all of this is that these effects are small. So, for that reason, we looked at repeated measurements in the same behavioral state to try and convince ourselves that they were real. This is an ongoing study where we're continuing to recruit and analyze the data, but this is kind of where we stand so far with what we're seeing. And then the last conclusion here is that if you look at mindfulness meditation and sleep, there's really no clear change in the cortical or the choroid plexus perfusion with sleep or meditation in the way that in the participants that we saw. That may be there, but it seems to be a less significant effect. The increased inflow in the fourth ventricle during meditation, so we saw some evidence of that, similar to what that Boston group showed in their nice sleep paper. And this inflow effect was anti-correlated with the salience network hemodynamic activity during meditation in multiple participants, but not all. And that work was sort of, or those findings were analogous to the prior work in stage 2 sleep from the Boston group that I showed earlier. So some of the things that I kind of think with this is the first, as you might imagine, is that it's really difficult to meditate and sleep in the scanner. And the rigor of these studies can probably be increased with improved metrics of meditation performance. And I think some of the reasons we're not seeing significant findings in some of these people is they don't do a good job meditating in the scanner. Just because you meditate every day outside the scanner doesn't mean you can do it well inside the scanner. So I think having better ways of controlling that, doing more people and whatnot, is probably important for these studies going forward. And right, just like I mentioned, is that lifetime experience doesn't necessarily mean that you're going to do a good job in the scanner. So we have methods for querying people about that and how they did, but it's still a bit variable. I also think, in talking with these participants, is that meditators, as people here know, can be highly heterogeneous. And what I started realizing is that meditation activity is often comorbid with lots of other conditions. And what I mean by that is people don't just meditate every day because they like it. They often meditate every day because they've had traumatic experiences. They have depression, they have pain, and the meditation is sort of a soothing or a therapeutic treatment for some of those conditions. And that's great, right? But what you often need to do is you need to also control for those other things that may be comorbid in these participants. So we do that a bit with our other imaging and some neurological assessments, but I think that's something that could also be done better in the future. So I think going forward, we'll continue to do this. I find it really interesting, and I think the future directions, maybe to assess different meditation practices. We haven't done that yet. Introduce meditation as an intervention. So I think this is something that I'm kind of interested in, is that it's difficult having people meditate in the scanner. But suppose you brought in people, you scan them, they were meditation naive, and then you had them go through training for the next several months, and then you scan them again, and you look at how that affects sort of more chronic changes in brain health and that sort of thing. That would be an easier study to do actually than what we did here. So I think that could be something interesting. You could also pair it in the setting of neurodegeneration to see whether things like proteins reduce after you do those interventions, similar to like a lecanemab or donanemab drugs that are reducing the amyloid now in Alzheimer's patients. And what I'd also like to emphasize is that the tools that we've helped develop should have wide relevance to the growing number of applications where neurofluid circulation is implicated. So what I mean by that is, if there are other people here that are kind of interested in doing this, we'd be more than happy to share tools or knowledge or whatnot, or if you want to send us people to scan, that would be great. So, I'm interested in hearing other comments or suggestions. That would certainly be welcome. And I will just end with the acknowledgement. So a lot of people go into making these papers successful, or this work successful, and I've listed them here. A couple that I'll call out, like getting this all going initially, was from Adam Stark and Caleb Hahn. That was a lot of work, along with Alex Song. Dave Vago is our sort of resident meditation expert. Bryce Keating, who I mentioned, has done the bold assessments. And then Colin McKnight is a neuroradiologist that reads the scans. Kieran Considine, who you're going to hear from later this week, is a clinical neuropsychologist. And Daniel Clausen is a neurologist. Killian is our deep learning expert. And we have lots of others who have contributed in different ways as well. It would be unsuccessful without all of the people on this slide. So I will stop there, and thank you for your attention.
Dr. Donahue, thank you very much for your inspiring, pioneering work that offers a great deal of hope, not only for the clinical applications, but also potentially in the future for some policy implications. In terms of questions, we have several that have come in, and I'm going to start with one that has to do with the rigors that you have upheld, which are quite inspiring, and will speak to the broad concern that there hasn't been enough randomized controlled trial data, as you summarized. What has the receptivity to your publications been?
Right. It's kind of interesting. We do a lot of work in patients with stroke, guiding treatments and whatnot, evaluating how those treatments work. It really has like very real, tangible impact on health, I think. When I go and give talks, nobody seems to care so much about that anymore. Everyone wants to hear about the meditation. Everyone wants to hear about meditation in the lymphatic system. I think those are red hot areas right now. So we've received a lot of attention from the work that we're doing, and a lot of interest in it. Yes, is what I would say. It is still very new for us, I would say, and I think the findings that I've shown today are probably about right, but I think some of the studies are still underpowered to make those conclusions, and we have to continue to get people in the scanner. But the story that's being told, I think, that sleep modulates clearance in the brain, and that you can simulate, or you can invoke similar properties with mindfulness meditation, is in focused awareness, is a very credible story, I think. Like, even if that's not true, I think you can listen to that and believe that it's the case. So we've received good, a a lot of reception from this, mostly from talks. It's still early, so we're just submitting our first papers on the meditation components, at least. So we'll see how that's received in the next year or so, I guess. But in terms of talks and whatnot, you know, everyone's interested in meditation, right? So we get a lot of attention for that. Yeah.
Thank you. That's good news. And you said that you're still recruiting. What is the best way for anyone who wants to reach out to offer to be a participant to contact your team?
Right. So the best thing is contact me.
Thank you. Have you considered focusing a study on practitioners who are practicing sometimes it's referred to as dream yoga, or from the traditions that really are consciously acknowledging that they are trying to influence their state of conscious experience in sleep and as well as in meditation?
Right. It would be very interesting. I am aware of that, and I've had some discussions about that. There's actually a review article that's in process where we will discuss some of those things. But right, so I'm not an expert in that by any means, but as I understand it, that there are certain practices where, you know, in very adept meditators with certain practices, the idea is that they obtain the restorative effects of sleep during these practices, and as a result, they sleep less, more or less. And, you know, I think that would, you know, I mean, that would be tremendously interesting to study, right? Like I would love to do that. The trick with that is finding those people, right? Like we're in Middle Tennessee, which is not the capital of the experts that do that. But, you know, and unfortunately where those people are, there may not be high field advanced neuroimaging equipment that's easily accessible either.
Your physics background, maybe you'll be designing the MRI machine that can be carried to those mountain cliff monasteries one day.
Yeah, that's what we need.
Yes. So following along in terms of study design, could you review, if we apologize if we didn't fully follow, the extent to which you monitored the participants' sleep health prior to being qualified for the study, or monitoring it during the study, especially during the sleep deprivation day, and what the person's activity was during sleep deprivation?
Right. So sleep deprivation, so we keep them up all night, and what they have to do, so they wear an actigraph, which sort of monitors their activity, and we can see whether they're sleeping or not. So there's an actigraphy monitoring just to make sure that they're not sleeping. And we have things that they do. We've tried different things and they're all fine. Like you call the grocery store every hour and ask them if they have apples on sale, and they come and leave us a message or whatever. So we do little things like that to keep them awake, and we monitor it with actigraphy. What we do now is actually much more complicated. So we now have Fitbits and this Fit Cloud thing where they can we have people wear that for a week before the scan, and we monitor their whole circadian rhythm to see whether that has anything to do with what happens in the scanner as well. But that wasn't done in this study. That's done in a new study that we're doing now. But yeah, to answer your question, we use actigraphy to make sure that they're awake all night. And that first sleep deprivation scan is kind of interesting. Like that tells you a little bit about what happens when you don't sleep for a day. And that's a little bit different than meditation, but that's an interesting thing that we're looking at as well. And then during the scan, you know, they wear this EEG cap, and we've got a neurologist that does clinical grading of those scans. We also calculate the delta alpha ratio, which is also a kind of continuous measure of sleep activity, and we look at how that correlates with some of these two. So a lot of that is not finalized yet. We're still deciding what the best way of doing it is. But for the data that I showed today, those are participants that have confirmed sleep from a clinical neurologist looking at the EEG recordings.
Your rigors and thoroughness are inspiring. Thank you.
Oh.
Pivoting to the field that you've emerged from, or are still very much in, about neurodegeneration, could you speak to something that I didn't hear you mention, which is the prevalence of people with essential tremor that is not considered Parkinson's or some other diagnosis, and whether the trajectory of your research could potentially offer an early detection screening technique that may be triggered by the presentation of essential tremor?
Yes, good question. So I work very closely with the movement disorders neurologist, Daniel Clausen. That's one of his major areas of work, actually, it's actually Parkinsonianism. So understanding how Parkinson's varies from essential tremor and what the different pathways are for that. I would say I am less of an expert. We do have a paper looking at diffusion tensor imaging, looking at fiber traction in the brain and characterizing which pathways are different between those two conditions. So I'm not totally sure. Essential tremor, is not a proteinopathy in the same way that Parkinson's is. So it wouldn't surprise me if these pathways are less relevant to essential tremor than they are to Parkinson's. But the safe thing to say would be that I've not made these measurements in ET patients, essential tremor patients. And it's a huge problem, right? I mean, I forget what fraction of the participant patients, individuals have ET over age 65, but it's huge, right? Like we all know people probably that have those problems. So we have not looked at that. It would be interesting. My gut feeling is that this stuff would be more relevant in Parkinson's than in ET, but I haven't done it myself. Yeah.
Well, you've done so much, and you're generous with what your work has been. Dr. Manus Donahue, thank you for weaving together your physics, your philosophy, and your commitment to health and well-being for all. We're grateful to you.
Yes, that's very generous. Thank you. Thanks for inviting me as well.